What is DNA?
In most basic biology classes, you've probably been told that DNA is a fixed set of nucleotides that codes for everything, a detailed instructions, or a map.
You have a gene for attached earlobes, a gene for straight or curly hair, a gene for calicoism, maybe not the last one. Through this idea, we learn that evolution is a slow change of DNA in a population from generation to generation. Mutations in the DNA of germ cells that create this change can be advantageous, detrimental, or inconsequential to the offspring. Then, through a process of natural selection, the advantageous genes slowly become more frequent. But why can't evolution evolve? It's not exactly the most efficient process. It takes loads of time and resources. And it takes an incredible amount of luck to get the right mutation at the right time. When something like the human genome consists of 3 billion base pairs, and mutations are as rare as 1 in every billion copies of DNA. You may have even heard that only 3% of our DNA even codes for anything, and the rest is just junk or non-coding DNA.
That junk may just be the solution to our evolutionary problem.
Barbara McClintock was breeding corn in 1944 when she discovered that the corn's color pattern was changing too rapidly from generation to generation to be the work of simple mutations. As she looked closer, she theorized that entire sections of DNA were moving from one place to another. The movement of these jumping genes, or transposons, was not completely random either. If it was random, one would expect to see them broadly dispersed over the genome, but in reality, they often land in a pattern, inserting themselves in specific sections.
This pattern begins to make sense when you start to think about what's actually causing the jumping. McClintock believed that the genes were pushed into motion because of environmental stressors.
Let's take an example from Todd Schlenk. If a population of fruit flies cannot digest a certain pesticide, but all the produce it has to eat has been covered in the pesticide, it would make sense for transposons to move to genes that could potentially allow the fly's immunity to the toxin. Sort of like a game of poker where a bad card can be replaced in exchange for a random draw of the deck. A lucky fruit fly gets a good card, and our process of natural selection allows the new combination of genes to be passed on and spread throughout the population, while less fortunate fruit flies die off.
This process of jumping genes allows an organism to react to its environment and pass on its DNA, rather than having to wait for the right mutation to develop.
Even though McClintock's discovery was not initially accepted by her peers, other scientists began to discover similar genes in organisms outside of corn. Today, 45% of the human genome is expected to be jumping genes. Jumping genes can tell us a lot about DNA. They help discover how complex a system like DNA can actually be, how individuals have a hand in increasing their offspring's chances of survival, and there's still more to discover.